Okay, Bob.
Thank you, George. Thank you very much for your still be here in the last session. So I know everybody is quite tired in the whole day meetings. So next, maybe 20 or 30 minutes, I will show you some application in capillary electrophoresis. So right now, just to let you know already about the next generation sequencing. Hang on. So sorry. Next generation sequencing is for ion proton and ion PGMs. For the conclusion, the PGM is suitable for small genomes, the less than 1 gigabases, and ion proton is should be fit with the bigger genomes, more than 1 gigabases. Anyways, two platforms can use in the transcriptome applications or ASEQ and ChIP-seq also. Another application is capillary electrophoresis. So right now we have a new ones we call 3500 and 3500 XL. These are 8 capillary and 24 capillaries. Actually, we know the capillary electrophoresis is gold standard. We use it for uh, confirm the sequencing, confirm the SNP, confirm the mutation from the next generation sequencing. And anyway, you can use this for the small region sequencing. So I believe that everybody in this room have been used or get the data from the capillary electrophoresis. So life technology have various platforms. Whether you would like to single capillaries or multi capillaries, life theory provides you and support you. For the single capillaries, we call 310s. And the 3130s, 3130 XL is for 4 and 16 capillaries. And the highest throughput, the capillary electrophoresis, because 2730s and 2730 XLs is for 48 and 96 capillaries electrophoresis. And the newest model is 75 XL, 75 series is for 8 capillaries and 24 capillaries. There are a lot of improvement in the hardware and also software. This is a little bit easier for user operating system. Everybody know about the cycle sequencing is the gold standard. Actually, it's very fast of next generation sequencing development. There are a lot of applications in the next generation sequencing. The cycle sequencing is also used for confirm the result from the next generation sequencing. Actually, they can use the cycle sequencing for close the gap and confirm the some regions that cannot be identified by next generation sequencing also. Actually, the common application for every user who have a capillary electrophoresis is for de novo sequencing and resequencing application. This technology can get you the data at high accuracies and very fast. You can detect the rare alleles, maybe 20% of the rare alleles, and you can get the sometimes if you prepare the high quality samples, you can get the rare allele frequency less than 10%. So let me show you another application from the capillary electrophoresis. That is the DNA fragment analysis. The common workflow for the capillary electrophoresis. The first thing, you need to amplify the particular regions by the fluorescent labeling primers. And then you take the fluorescent labeling PCR product and then mix it with the size standard. The size standards also have this is the standard fragment and labeling with the different fluorescent than the unknown fragments. Mix it up and then load to the capillary electrophoresis. Live COD also provides you the software we call GenePlus. The software will calculate for determine the size of the unknown fragments. Actually, this capillary electrophoresis can detect the multi colors. This means you can multi primers that label with different fluorescence and mix it up and run together in a single capillaries. So I would like to show you some application in the fragment analysis for rice research. The first is SNP genotyping. The second is ISSR genotyping, is another polymorphisms. And finally, I will show you the AFLP application genotyping in capillary electrophoresis. The first SNP genotyping. Live state we have a kit we call a snapshot. This kit is based on the single base extension labeling chemistry. The workflow, you just amplify the particular region that have the SNP, and then you decide the primers that located behind the SNP position, and then mix with the labeling chemistry dideoxynucleotide. After PCR reaction, you take the PCR product, mix with the hydride formamide and size standard, and run to the capillary electrophoresis. After you get the data, you put the data in the software we call GenePlus. The software will call you the alleles. You can see the difference. You can see the different colors. The different colors represent the different dideoxynucleotide. This kit allows you detect up to 10 SNP per run. So this is an example publication using the snapshot based on sequence-based primary extensions. This paper displays the genome, display the full SNP from the competitive sequence analysis. They use the 16 capillary genetic analyzers. This paper can identify the new markers for virus-resistant genes in oat. The researchers conclude that they can run more than 700 samples per day after you purify the PCR product and prepare the PCR reaction mix. Because of we have various platforms in the capillary electrophoresis, the number of SNP per day starting from 400 to 40,000 SNP per day. They calculated based on 10 SNP per capillaries. So this is low and medium throughput SNP genotyping assay right now. And another application in the fragment analysis is for microsatellite ISSR analysis. Intersimple sequence repeats, or ISSR, is a polymorphism between the microsatellite regions. The ISSR PCR is very fast and inexpensive genotyping assays. You can use this method for characterize genetic relationship in the among population. Various primer designs. You can design the primer in the microsatellite regions. Whether you can design the primer have a 5' flankings and also 3' flankings. To increase the specificity, you can amplify particular regions in genomes. Actually, there are a lot of publications for using the ISSR markers to study the some stress condition. This paper show ISR-based genotyping to study the relationship among rice varieties. The second publication showed the application of ISR markers to typing yeast that isolates from the rice production. Finally, this paper showed the ISR marker for identify the fungus that cause of plant troubles. This is a particular results from the ISSR PCR and then loading the regular gels, agarose gels. You can see it's quite difficult to interpret that. Some researchers said that if you run ISSR PCR product in the agarose gels, it's quite poor reproducibility and take your time consuming data analysis part. Life Technologies developed the workflow for studying fragment analysis of ISSR methods. They also have the same workflow with the traditional ISSR PCR. The first thing, the exception. Actually, the difference is the PCR step. If you would like to study the fragment size in the capillary electrophoresis, you must need to decide the primer and have the fluorescent dye in forward or reverse primers. And then take the PCR product, mix together with size standard and run to the capillary electrophoresis. After you get the data, you put the data to the GeneMapper software. You can see the example of the ISSR from the capillary electrophoresis. This figure shows the different results from the each plant species. You can see very clear difference in the ISSR pattern. So the last application for fragment analysis we call high coverage gene expression profiling or HiSEP. This is also based on the amplification fragment length polymorphisms based gene expression profiling assay. The advantage of this method is require no parallel sequence information and has a reduced rate of false positive. And you can detect of both coding and non-coding regions. And you can detect in every transcript. Furthermore, after you run the HiSEP analysis, fragment of interest you can purify and clone in the gels, and then sequence to identify the region of interest. This is the workflow of the HiSEP. After you correct the mRNA, you synthesize the double-stranded DNA by using the poly-T and link with the biotin. And then digest with specific region enzyme. Normally we use MSP1. And after that, like it with the specific adapters, and then purify the particular fragment by using the magnetic bead linked with peptidin. And then digest with another region enzyme. Normally they use MSE1. And they like it with another adapters. Finally, they amplify with the specific primers. Actually, the primers will label with the region is labeled with the fluorescence. Amplify it, take it and pull it with the hydroformamide and size standard and load to the capillary electrophoresis. This is the example result from the high-step technologies. In this experiment, they have two correction mRNAs, and they use the different fluorescent labeling primers. The first label with farm, and another label with rocks, and run together in a single capillaries. You can see the level of the fluorescence represent the mRNA levels in different species. And after that, the researcher can take some PCR product running the gels and cut the gel and cloning and sequence to the Sanger standard method to identify what is the fragment, what is the transcript. There was the papers published in 2005. They used high-step technologies to study the gene expression during rice seed development. The researchers conclude that these technologies can identify more than 70% of the mRNA. They use the mRNA sequence database and digest with the in situ PCR, and they found that about 40 and 70 base pair size of the fragments can be digested with the double enzyme. So furthermore, the researcher can identify the novel genes that involved in the starch synthesis pathway. The researcher conclude that high-step allow to study in the expression profiling in rice. And they said these technologies lower cost than microarrays and DNA chip assay. And they said this method is very easy and get more data from plant species. So this is some application from fragment analysis. If you're interested in de novo sequencing, small target resequencing, confirm of next generation results, SNP confirmation in low and medium throughput, methylation analysis, QTL mapping, linkage mappings, and also copy number variations, microsatellite analysis, AFLP analysis. Which instrument is best for your plant research? Exactly, maybe the capillary electrophoresis. Just remind you, the capillary electrophoresis is the gold standard for the DNA sequencing and the fragment analysis. Thank you very much.